Ha ha ha, suckers! I got you to come back for part 2. Alright, so part 2, we're going to leave off a little bit with where we left off, which was the history of genetics. We're going to go into a little bit more about what Mendel studied and what Mendel found throughout his studies. Okay, so we talked about kind of the brief overview of genetics in part 1, as well as just a brief history. So make sure you go back and read in your lessons to fill in any gaps, because I didn't go into that in detail, because you guys can definitely read through that. Alright, so in going on, here's where we left off. Here's one of the things that Mendel found, the principle of segregation. I don't want these terms to kind of make you go, oh my gosh, I'm never going to get this genetic stuff. Because really, when you break it down, it's actually fairly simple. Okay, they're simple concepts, and they will make a little bit more sense as we start to do more with them. So the principle of segregation says, for any particular trait, the pair of alleles of each parent separate, and only one allele passes from each parent to an offspring. Okay, so let me kind of simplify that a little bit. First of all, I want you guys to know what an allele is, because you're going to hear that term a lot. An allele is a genetic term, and it's basically another fancy schmancy word for gene, but you're really just taking one of those pairs of genes that are found on the chromosome. Okay, so really, if you want to reword this, for any particular trait, being like curly hair versus straight hair, the pair of genes of each parent separate, and only one gene passes from each parent onto an offspring. So gene, again, another name for that is kind of an allele, and you're going to hear that a lot. I'm going to say that a lot with some of the definitions that we have. So one allele passes from each parent onto an offspring. So really, they segregate. Either you can use the term separate or segregate kind of interchangeably when it comes to this principle, okay? So basically, one gene is going to separate from the other and pass on to the offspring. So pretty easy, right? Principle of segregation, genes separate. Okay? Hopefully that makes a little bit of sense. Now, let's talk about, when we talk about the law of segregation, we also talk about a monohybrid cross versus a dihybrid cross. A monohybrid cross is cross-pollination involving only one trait. So in that video that you guys watched in part 1 about Mendel, he first did one experiment where he just did a monohybrid cross. He involved only one trait, which was basically, is the pea round and smooth or wrinkly? That's one trait, smooth versus wrinkly, okay? The dihybrid crosses, when you get into that, are going to examine the outcomes of two traits. So maybe you throw in, is it yellow? Or is it, like, basically we're talking about color versus texture, okay? So I don't want to confuse you guys when I say the monohybrid. We're really just talking about one trait being texture. Is it wrinkly or is it smooth? But a dihybrid is going to actually test whether it is wrinkly or smooth, so the texture, and maybe the color. So you're taking two traits. So pretty easy. Mono means one, di means two. So monohybrid, one trait, dihybrid, two. Okay? So hopefully that makes a little bit of sense, and we're going to look at that when we talk about good old Punnett squares. Let me show you this quick picture here, though. This is just an example of the law of segregation, meaning that these are kind of the parents up here, okay? And basically you have one of these alleles, basically one of these pairs of the genes, that is going to go one direction, basically to an offspring, and the other one might go a different direction. Okay, there's not really any rhyme or reason to it, but that is the law of segregation, that basically those alleles will separate and go to the offspring. Okay, so hopefully that gives a little bit of a, kind of a picture for you guys. So let's move on to Punnett squares, and you guys have an activity or an assignment that you're going to be doing with Punnett squares. I like Punnett squares because they kind of simplify things, and if you guys are kind of ever so slightly good at math, you don't even have to be good at math, so I don't want to scare you away if you're not good. But it's simple multiplication where you can figure out genetic traits, and that's with Punnett squares. So Punnett squares are a visual for showing probability. Probability is, what are the chances of my child having blue eyes? So that's a probability. Is it 25% chance? Is it a 50% chance? Or 100% chance that my kids will have blue eyes? That's probability. Okay, we're using percentages. So the next thing that a Punnett square does, but before then, let me show you my cool little tattoo picture. Not my tattoo. Look, I promise. Nope, nope. I'm not that much of a science freak. I would never get a Punnett Square tattooed on my wrist, but I thought it was kind of funny. This is a Punnett Square. We'll go into a little bit more what that means, but someone is a freak about Punnett Squares. Not I. I like them, not that much. So Punnett Squares show all possible gene combinations from a cross of parents whose genes are known. Obviously, you have to know those genes. And really, it's pretty easy to know those genes because a lot of them, not all of them, but a lot of them you can see, okay? I have hazel eyes. That is a gene that's known. You can see that, okay? My kids, one kid has brown eyes, the other kid has blue eyes. So really, we could also use my husband's genes, he has brown eyes, to figure out what's happening in order that we received a child with blue eyes and a child with brown eyes, okay? So let's kind of talk about that. I'm going to show you guys two examples of Punnett Squares. So this one right here is basically showing you that when people come together and make babies, yeah, let's see what are the probabilities of basically having boy or girl, okay? A male, you have an XY chromosome, okay? So basically when we do a Punnett Square, depending on how many traits we're using, okay, we set up a square. The easiest is basically a 4 squared square. 4 squared square. So the male has an X chromosome and a Y chromosome, where the female has 2 X's. So all we're going to do is cross multiply. So X times X gives you an XX, that would be a female. X times Y gives you a male. X times X gives you a female, and X times Y gives you a male. So obviously you're seeing that 50-50 chance of a male or female. Pretty easy Punnett Square, right? Okay, here's another one dealing with something different, okay? Alright, this is talking about eye color, okay? So we're looking at Carrie here, who has big B, little b, which you'll understand a little bit more, okay? And then you have Rusty, who has 2 little b's, alright? And if you, I'll get into the whole dominant versus recessive, but if you multiply these, and what we're saying here is that brown is dominant, that's why it's a big B. Blue is recessive. A dominant trait, which I'll get into, I'm kind of jumping ahead here, is always going to show up over the recessive trait. So if there's a big B in the equation at all, that person's going to have brown eyes. So let's just do the whole basic cross multiplying again. Little b times big B is going to give you big B, and we always start off with the big B, just so you know. Big B, little b. Little b times little b gives you little b, little b. Here we got big B, little b again, and then little b, little b. So with this set up, where we know Carrie has brown eyes but is still carrying the recessive trait for blue eyes, and Rusty has blue eyes, clearly, because he's got 2 B's, that means the offspring has a 50% chance of having blue eyes, because this is blue eyes right here, and this would be brown eyes. So 50% brown eyes, 50% blue eyes. Now again, if this Punnett Square stuff is all new to you, I promise we'll practice more with it. Just let it sink in. But basically we're doing kind of a cross multiplication. You have to know this genotype, which we'll get into what the genotype is, but you have to know these things before you can actually figure out what the probabilities are, okay? But this is a basic Punnett Square. Now you may have different, a long list of traits that you want to check out, and I'll show you what a Punnett Square looks like when you're looking at more than just, say, eye color, for instance, okay? So before I show you that picture coming up, let's talk about the principle of independent assortment, okay? Again, it sounds pretty technical, but I promise it's really not that bad. What it is basically different pairs of alleles, meaning genes, so different pairs of genes, are passed to offspring independently of each other. So really, you're not going to have a bunch of genes that kind of work together, like, hey, eye color, how about if I stick with you? I'm hair color. No, doesn't work that way. Hair color basically is separate from eye color. All of the genes separate independently of each other. They are passed on to your offspring independently, okay? So you could have one child that has curly hair and blue eyes, and your next child could have straight hair and brown eyes, okay? They are always independently passed down, okay? So hopefully that kind of makes sense, even though the independent assortment, you're like, gosh, why do they have to name things so crazy? But really, that's all it's saying, okay? Now, I want to show you guys, this is more of a dihybrid cross. What we looked at, the Punnett Squares before, dealing with how many, what's the probability of a male versus a female in the offspring, and also eye color. That's a monohybrid cross. That's one trait. We looked at sex and we looked at eye color in both of those Punnett squares. Here's a dihybrid cross. Now remember I told you, so in the example of the peas, and this is what we're looking at right here, we're looking at color and we're looking at texture, okay? So if you notice, you have, first of all, basically all of these things that, you know, round and yellow, round and green, wrinkled and yellow, wrinkled and green. And here are the different genotypes for that, okay? The genotypes are basically the genes that you can basically cross-multiply with the parents. So these are the parents. You always find the parents on this side and this side. Here are the parents' genes right here. You have big R, big Y, big R, little r, big Y, and little r, little y, okay? And the parent has the exact same things up here. When you cross-multiply, now you have 4, basically, that you're looking at. So obviously you can tell which are, well, you can't tell because you don't know a whole lot about the dominant versus recessive, but I did tell you that dominant are going to be in capital letters. So obviously when you have big R, big Y, you're dominant in both of those traits, meaning round and yellow, okay? So I don't expect you guys to be able to look at this dihybrid cross and understand it, but it basically is just a little bit more intensified than that monohybrid cross, where now you have 2 traits you're looking at. You're going to cross multiply, so that's why you get big R times big R, so you have 2 big R's. Big Y times big Y, so you got 2 big Y's. Now look at this one. If you cross multiply these two, big R, okay? Big Y, little y. So there you go. It's very simple math when you think about it. And this gives you probabilities of what is what, okay? So again, dihybrid cross, testing 2 different traits. We'll come back to that a little bit, but just understand the gist of it. Remember, this is biology. You'll get this a lot more as you go into college and study this more in depth, okay? But that is a dihybrid cross. Remember, that has to do with the independent assortment, where basically those alleles are going to go to the offspring independent of each other. So color and texture are not going to go hand in hand. It's kind of like, hey, whatever. All right. Now, genotypes and phenotypes. Here's where I maybe should have explained this a little bit more, okay? But the cool thing is this will all kind of make sense. So genotypes and phenotypes, this is a good thing, good definitions to understand. So the expression of a genotype, okay, so genotype, if you recall, and I mentioned it, not that you're going to know for sure, but a genotype is, say for instance, I had blue eyes. My genotype would be little b, okay, that's a genotype. But the expression of a genotype, so basically what you see, is the phenotype. So I think of pheno as like a physical trait where you can see it. You can see what color my hair is, reddish, brownish, whatever it is. You can see what color my eyes are, kind of, maybe not in this lighting, hazel. That's the phenotype. But the genotype is a little bit different. The genotype are actual what represents that in the form of letters, because that's how we do Punnett squares and figure out probabilities. So the genotype is the actual genes. The phenotype is, hey, how's that expressed? How's that look on me? Do I have curly hair or do I have straight hair? Okay? So for example, the phenotype would be hair color, weight, or the presence or absence of a disease. Okay? The genotypes are the genes that code for that. So I want to kind of back out here a little bit. This picture over here, if you guys see, genotype codes for phenotype. So genotype are basically these genes here that are found within the strands of the DNA, okay? And they code for what you're going to see on the outside. The color of a flower, the shape of the leaves, all of those things would be the phenotype, okay? All right, here's another example. So the phenotype would be blue eyes. The genotype would be little b, okay? That's recessive trait. And then a phenotype here would be brown eyes, because you can see this person has lovely brown eyes. Where the genotype could be one of 2 things. It could be big B, little b, or big B, big B. Remember, since this is a dominant trait, that if there is a big B in the equation anywhere, that person's going to have brown eyes. So it could be big B, or big B, little b. But in order to get blue eyes, you have to have both of these recessive traits. So blue eyes are a little bit more rare. That's why people are like, ooh, blue eyes, because you don't see them quite as often as, say, brown eyes, okay? So that is genotype versus phenotype. Here's a little video to kind of fill you in on any
Your
gaps.
observable traits, also known as your phenotypes, result from the interaction between your genes and the environment. This interaction begins in the womb and continues throughout your life. Differences in some phenotypes, like height, are determined mostly by genes. If you had short parents and grandparents, you probably don't tower over your peers, though environmental factors like nutrition can have some effect on your height. Genes also play a role in whether you have a healthy body weight. But diet and exercise can profoundly affect how much you weigh. How genes influence your personality is less understood. You can now learn about over 500,000 SNPs in your genome all at once. And every day, scientists are learning more and more about how some of these SNPs affect your phenotype. Getting to know your genome can help you understand a little better why you are the way you are, and in what ways you're similar to or different from your family, friends, and neighbors.
So there's a couple of terms in those videos. One is genome. Genome is kind of like a map for all of your genes, and we all have different, well, I should say it's kind of a map of all the genes that exist, but we do have different genomes within ourselves, okay, our different maps, so to say, that make us who we are, okay. SNPs, I'm not going to get into those because that's a little bit higher level genetics, but if you want, go research it because it's kind of fascinating when you think about it. So dominant versus recessive, which I've already brought up a little bit. Let's look at this picture here. The effects of a dominant allele, B, big B, are seen even if it's present with a contrasting recessive allele, little b. So when we're talking about colors of cats, brown cats, basically big B, is their dominant trait, that even if it's paired with a little b, it's going to be a brown cat, okay? Big B, little b, brown cat. Gray, recessive, little b, has to have both of those recessive traits in order for that to show up, okay? So obviously you see that little b is being masked by that dominant trait, okay? So that's kind of the picture, but a dominant allele is expressed even if it's paired with a recessive allele. Remember that. Anytime there is a dominant trait, that will be expressed, regardless if there's a recessive trait with it, okay? And then, a recessive allele is only visible when paired with another recessive allele. Okay, that's why some recessive traits you don't see a lot in people. There's a lot of different recessive traits that, you know, kind of, hey, do you have this kind of trait? Do you have this kind of trait? Well, that's recessive. So maybe not a lot of people have that trait. So it's kind of cool. Kind of cool to see where you are. So, last thing that I want to bring it all together is with a song, and it's a cheesy song because it's good old Bill Nye, but it's pretty funny. So let's tie this all together with one last song, and then I will say goodbye. Go get it, Allison Jeans. Ow! No, anyways, I want you guys to know that we will get to, because some of you guys might be asking, okay, you've touched on brown eyes and blue eyes, but what about hazel eyes? And curly hair and straight hair, but what about wavy hair? So stay tuned for section 2, because we'll learn more about that stuff. All right, until then.